did her PhD in Brown University, where she was a graduate Ostrich Fellow. Before that, she did her master's in aerospace engineering at the Iowa State University and a bachelor's in mechanical engineering at the University of Akron. She has a wide variety of expertise in fluid mechanics, so you'll hear a little bit today about her work on shrimp as a model organism for bio-inspired underwater vehicles, and she'll also share some of her work that she's been carrying on at UTIG. She's also won a few innovation awards for her research, including work on the fluid mechanics of breastfeeding and also looking at improving swim teams and thinking about ocean tracers. And we're really delighted to welcome her to UTIG and to the seminar. So Sarah, please take it away.
Thank you. Okay, Krista basically did the first 5 minutes of my presentation, which was introducing myself. So thank you. So I kind of got you all here on the premise of shrimp. I am going to kind of switch it up a little bit. And for the last 20 minutes or so, I'm going to talk about the work I'm doing here. But for sure, as promised, there's plenty of shrimp. If this is not enough shrimp for you, just send me an email. I'm happy to talk shrimp for 3, 4, 5 hours. So you're always welcome to do it. The video you see on the background of my slide is of the small shrimp Halocaridina rubra. And if you saw the little shrimp that I had in my office that are now in Elizabeth's office, this is exactly what you saw. These are small shrimp native to Hawaii. And they're some of the subjects that I use in my research. And you'll see that they swim. I think if we've seen shrimp swim, we know they swim really funny, where they have their legs beating one after the other. And that's exactly what you see. So their backmost leg will start beating, and then the next one, and the next one, and the next one. This delay is a phase lag. And this method of swimming is called metachronal swimming. Shrimp use this locomotion method really successfully to navigate all different sorts of environments. So sort of as promised, I will talk about shrimp for sort of the first half of this talk. And I will mostly talk about the development of a shrimp-inspired robot. So if I could, it started in 2022, where I developed that first little leg, and now there's a whole team working on this. And in 2025, we sort of have an independent robot that can swim around in the pool and can do all sorts of tricks. So it's been evolving really quickly. It's all based on shrimp, and you can see various shrimp swimming. So at the top, you have a top view of shrimp. You're going to notice that their legs intermittently appear. This is because during the power stroke, they're going to amplify the area of their legs to make sure that there's enough area. And then during the return stroke, they tuck them in to reduce drag. And I'll go more into this. I just wanted to kind of give you an overview. The really long-term goal of this project is that we can make pods like you see in this figure at the bottom. We can pack pods with hundreds of these little shrimp robots that we can send to Europa to explore under ice dynamics. So like the maybe 20-year vision is what you see that right there, where you have a shrimp-like robot that can do all these things, has a high-definition camera, a lighting, a laser scanner, and all these other instruments. And then for the second part, I'll talk about the work I'm doing here. So I'm mostly an experimentalist, and 99% of what you'll see today are results from experiments. And so I'm doing experiments here, and I'm doing them mostly in the flume. The top image that you see there is the flume. It lives in the building right next to this one, the Center for Water and the Environment. It's a 20-meter flume, so really large. And I'm using that to understand sediment transport and reduced gravity. And like Krista said, I'm also working on these geometry-dependent tracers, for which I got an innovation award here with Benjamin. And I'll talk about those as well. So our motivation for looking at shrimp as model organisms comes from the need, really. If we look at this plot, we have maneuverability on the y-axis, and then we have spatial coverage on the x-axis. You really have a trade-off You can have a really maneuverable underwater vehicle, such as an ROV, that is going to have very low spatial coverage, or you can have low maneuverability and high spatial coverage with a glider, for example. We think shrimp sort of live in this really, really great area where they have really high maneuverability and also really high spatial coverage. And we think this is because, first of all, when we talk to their maneuverability, you can really see that video. That shrimp is following a little piece of food, and you're going to see it turn around. And if you look at shrimp, they can do all sorts of tricks in a very short amount of space. So they're extremely maneuverable. And they're also globally distributed, which means they live in all sorts of environments from coastal to the open ocean. And they can be found from really the north all the way to the south. So we think they're a great model organism because they can swim large distances, which means great spatial coverage, and they can do all sorts of tricks, which means great maneuverability. And shrimp are not the only animals that swim in this matter, metachronal swimming. This locomotion method is distributed across a range of scales and Reynolds numbers. So at the top, you'll see a small paramecium. It's around about 100 micron. And you'll see all these cilia sort of beating their little hairs. This video shows a calm jelly also using metachronal swimming. And then at the top, you have a mantis shrimp and Hawaii shrimp. They all swim metachronally. So this tells us that there's some sort of ecological advantage to using this propulsion method in this area of sizes and range. So when we're talking about the Reynolds number, it's a common non-dimensional number in fluids. It is the ratio of inertial to viscous forces. If you have a very slow swimmer, sort of like a bacteria, you're going to be in a very low Reynolds number where viscosity governs the motion. This is also sometimes known as Stokes flow. If you are at a high Reynolds number, for example, whales swim at high Reynolds numbers, you can discard viscosity and you can just focus on inertia. And these are sometimes known as potential flows. With shrimp, we're in this intermediate regime where the Reynolds number can be from 10 to around 1,000. And this means that we cannot simplify by discarding either viscosity or inertia, and we must consider both. So this is a really interesting case of swimming. The Reynolds number for shrimp also has two different magnitudes. When you look at the appendages only, you can be at a low Reynolds number between 500 and 1,000, but when you look at the body, you're going to be a much greater Reynolds number, which means different physics are contributing to the forces acting on this body, which makes it really interesting because you have a scaling problem within this one singular shrimp. And so they swim metachronally, like I said, where they have one leg beating after the other. And they also modulate the profile area of their appendages, which means that during the power stroke, they completely open up their appendages, and during the return stroke, they tuck them in. This means that during the power stroke, they're producing drag-based thrust. That just means that we're producing drag in that direction, and our force is going to propel us forward, sort of like when we swim. And during the return stroke, we're tucking it in, reducing drag as much as we can, and then this generates net thrust that allows the shrimp to float. Like I said, I'm an experimentalist. I use one method in particular called particle image velocimetry, and I thought I'd just introduce it if you are not familiar with it. So with this method, you can have something in the water. You can have some fluid structure interaction, shrimp swimming in the water. You use a laser to illuminate particles that we see the water with, usually glass particles or plastic particles. And then we use a camera to record this. this is what an image looks like if you're not using a black and white camera. You'd have a red laser sheet. You see all of the little specks in the water. Those are glass beads. And then you have your animal in there if that's what you're looking. Usually we do use a black and white camera. And the two images that you see right there are from the black and white camera. And those are two subsequent frames. And what we're going to do is we're going to pick a window, which is going to be our region of interest. And we're going to track these particles within this window. And in subsequent frames, we're going to find the displacement of these particles, right? So knowing the displacement and knowing the time that has elapsed between each frame, we're able to get this local velocity vector. So what it looks like if you have some animals moving in your flow, you're going to see all these white particles. In this image, you have a mussel and some barnacles that live on top of this mussel. And you see all these particles moving around in the flow. So you get your displacement, your time interval, you get velocity. Then you can have all your velocity vectors together. And this is what that looks like. You sort of see very well the inlet, and you can also see the barnacles filtering as they eat. And then from the velocity, using the curl, you can get vorticity. So the blue and red colors are going to be your swirling or the vorticity in the water. The more intense the color, the higher the vorticity, and you have two different colors because you have rotation clockwise and rotation counterclockwise. So this is the method that I use most prominently. If you think this is useful for you, please reach out to me. I'm always happy to set up experiments. Probably too happy. And now I'll talk about the robotic platform. Al, the shrimp whisperer, sells calendars, by the way. He does not pay me to say this, but I buy his calendar every day. So now I'll talk about the robotic platform that I started to develop, and it's called the pleopod. So I want to show you what it looks like right away. This is one single appendage of one shrimp leg. The QR code takes you to the file that you can 3D print to make your own. And this is going to have all sorts of complications, as you can see. In the front-facing video, you're going to see what I talked about with this modulation of the profile area. This appendage that comes in and out. And this is passive, so this motion is only due to the fluid structure interactions. There's really nothing governing this motion. From your side view, you're going to see these two sections. The proximal section is called the protopodite, and then the distal section actually splits into two, and you have the exopodite and the endopodite. This motion that you see on the side view is controlled by two circles. So this is what it looks like, and this is how we got there, sort of by getting inspired by shrimp. So there are several angles that modulate the kinematics of shrimp. Much of this talk now will be just Greek letters, so stop me at any point if you don't remember what a Greek letter means. So the two main kinematics that we have are alpha and beta. These are the ones that you see on the side view. So these controls are the proximal and distal section of this appendage. And because they're so important, we wanted to have them active when we do so through a gear train. So we have two circles at the top. We have a gear train that goes through. The gear train continues through the proximal section and actuates the distal section. We managed to get quite close to what real shrimp are doing. So the black dashed line is real shrimp, and the blue and red lines are for this single appendage. Like I said before, this modulation of the area is really important. So we wanted to capture that as well. And we do so with this appendage that is passive and is ruled by angle gamma. So gamma is passive. It hasn't really been characterized in shrimp because it's quite difficult to get these values for shrimp. But I'm plotting what it's doing right there. And like I said, this is passive, so we're not controlling it, but it's still quite replicable. The gray area is the standard deviation. And then there was also this small detail that we identified, which is this, when you have shrimp going, their appendages don't seem to be parallel to each other. The little endopodite seems to be sort of perpendicular to the direction of the swimming, but this one that comes out really isn't. It's sort of at an angle. So if you had this, you'd have this angle. And we noticed this and we thought it was important to include this. We weren't quite sure what the difference was going to be in terms of the fluid dynamics, but we wanted to include it as well. So we did, and this is fixed into the design. So we designed the part this way, it's 3D printed this way, and then we add it to the robot. No one's really looked into this, so we're lucky to be able to do it from our lens first. And now that we have our methods, our PIV methods, and that we have our robotic platform, we can start answering questions about this system. And the first question that we wanted to answer was one of force balance. So shrimp produce drag-based thrust, which is drag in one direction means thrust in the direction of swimming. But they're also negatively buoyant, which means that if they're not generating lift all the time, they're going to start sinking. This is really not efficient. So there has to be a way in which they generate lift that allows them to swim forward, still put as much force into swimming forward as possible, but also remain at the same height in the water column. So this was the first question we wanted to answer. And I will give you the answer right away. Although it took maybe like 3 years for us to get the answer, you get it right away. It's very expedited. So what you're going to see in these videos that I'm showing you is our PIV images like the ones I showed before. The laser is where that red line is in that video. So you're only intermittently going to see this appendage coming in and out. And the camera is on this side where I am. So you're going to see the side view into this appendage. I'll show you the raw images first. You see this appendage coming into the plane. You're going to see these particles moving. And here it's a little bit difficult to understand what's happening, but if we no? Might have deleted all the videos. Okay, I do have the same video right here, so I'll show you that. Okay, so the top video that you see there is exactly the same as we were seeing before. You're going to see these particles going. Then if we just look at the top video so we don't spoil the next slide, please. If you look, now we're going to see the velocity field, and you're going to see these large vectors around the appendage with red coloring, which means higher magnitude of the velocity. But what really is going to show you what I'm talking about is this vorticity plot that's coming next. And you can see these two vortices with large magnitudes generated on the top of this appendage. These vortices are known as leading edge vortices, and they're most commonly used by insects to fly, which was a really interesting discovery for us because we were really not expecting this, as shrimp are known to use drag-based thrust and not lift-based sort of lift, yeah. And so this was really interesting and was something that we wanted to look more at. And the way this leading edge vortex works is by creating a suction pressure on the top of this appendage. And as it does, it generates lift upwards. So now we'll go through this again, but with a different perspective now. Now we'll look at it through the ecological perspective. If we want to compare two shrimp and the differences between them and understand where ecology comes in, we can do so using our robot as well. So here I'm doing exactly that. I'm going to be comparing two shrimp. We have the kinematics of their appendages for both. And the first shrimp, so the top one is krill. They live in the Southern Ocean around Antarctica. They are mostly pelagic, and so they swim for 100% of their life. The bottom shrimp is marsh grass shrimp. They live in the marsh grasses in coastal areas, and really they use their legs mostly to help with their walking and not so much for swimming. Because they're benthic, they don't really need to generate a lot of lift. They're just kind of at the bottom of the ocean. And we're going to see a very clear difference between the resulting fluid dynamics from the kinematics of their swimming. So this plot shows you the angle versus the time of this cycle. And you will notice right away that krill, which is the blue line, have a much greater amplitude. Marsh grass shrimp have a much smaller amplitude of their beating, and this is going to be reflected in the fluid dynamics. So we'll look at both videos now. And these are at the same time, but you're going to see at the top video, this exopodite is going to come into the plane much sooner, because with higher amplitude, there's more forces going in that direction. So you see the raw images. Now we're going to look at the velocity, and you can once again see the comparison. You're also going to see much greater magnitude of the velocity at the beginning of the cycle for Isoperba, for krill. But then when you look at the vorticity, you will see a huge difference. So for krill, we're going to see those really two large vorticity areas. And when it comes to marsh grass shrimp, you really don't see that. And when you do, this appendage is already fully vertical, which means it's actually producing thrust and not lift. So this is one way in which we're able to use this platform to understand not just the engineering part, but also the ecological significance of difference between species. So I did show you what we're doing with a single appendage, but I also wanted to show you where we're at now. And that's the little robot in its current iteration. As you can see, it doesn't matter how deep you started off with, it'll come to the surface because it's generating so much lift. This turns out to be a disadvantage for underwater robots because you really want them to be positive. You really want them to be positively buoyant, because if they stop working, you want them to resurface so that you can recover them. So we're still thinking how we're going to have a negatively buoyant shrimp be positively buoyant. Questions? Concerns? Okay, like we saw before, one really important thing that we focused on was this trade-off between lift and drag. How has this happened? Can we further understand this mechanism and take advantage of it so we can use it in our robots? So when we take a closer look at these two bioramus appendages, we have the endopodite and our exopodite, and the shrimp is swimming through, we see that there's actually an angle of attack between this exopodite and the flow. This is the same way airplane wings work to generate lift. So it's really an interesting mechanism that we wanted to study further. And we're going to do so with a simplified robotic appendage that you see on your right. That's going to have two main angles that we need to keep track of. That's gamma, that's once again the angle between your endopodite and exopodite that extends this appendage out of the plane. And then the other angle is zeta, also named the cupping angle. And this is the angle between your two appendages. So if they're parallel, zeta would be 0. If they're perpendicular, zeta would be 90. And you kind of have a range in between. So we tested this. And the first thing we did was, let's just run this appendage in the tank and track the motion of this appendage that's allowed to go in and out. So in this plot, the colors are going to be zeta, which is the red angle in that side, and your y-axis is going to be gamma, which is the green angle that you see. And you can see that there's a range of different kinematics for this gamma throughout the cycle for different angles. I'll highlight two cases, the case of 0 angle and the case of 40. And it's going to be very clear to you when you see these videos that there's a huge difference. Remember that this is passive, so we're not controlling anything. It's all controlled by the fluid structure and relations. For shrimp, they want to maximize the area of their appendages during the power stroke, and they want to minimize this area during the return stroke. So it's advantageous that this movement happens as quickly as possible once you begin your power stroke and ends as quickly as possible as you begin your return stroke. That happens to occur at an angle of around 40, which is very close to what we measured in shrimp to be around 37. So this is a really cool finding about how this angle can really help them modulate the generation of thrust and drag and also lift. We can further characterize the trade-off between these forces by measuring them directly. So we once again use this little appendage. The angles are the same, gamma and zeta. I just wanted to remind you. And this plot has the force and the different zeta angles on the x-axis from 0 to 80. That's the range we tested. And you see that lift increases as we increase in this zeta angle, and thrust diminishes as we go through this zeta angle. So you can very clearly see this trade-off. We think it looks something like this. When you're looking at it, you have your blue force would be your total force, and then this force decomposes into two forces in the lift and drag direction based on this angle. And of course, this is a really, really interesting result that we got to understand, but doing experiments can only take us so far. So really, we wanted to find a mathematical model that could allow us to predict lift and drag based on this angle for any Reynolds number for any parameter that we want to vary. That seems much easier than doing a million experiments, although not as fun. And we don't want to make it too complicated. So we want to base this model off of the very simple kinematics that we're able to extract from shrimp swimming. And those are going to be just the four main angles: alpha, beta, zeta, and gamma. So can we use these four angles to estimate the forces on these appendages and get a large parameter? We're going to try, even though this is a very 3D motion, we're going to use a 1D equation that is going to add up inertia and drag only. So we're going to have these two forces. The equation looks like the one on top. So the first term there is the force due to inertia. Cm is your coefficient of inertia, rho density, v is the volume, and dv dt is the acceleration. Then your second coefficient there is the force due to drag. Cd is the drag coefficient, v is the velocity, a is the area. We have rho once again. We're going to use this very simple equation to see if we can estimate the forces on that very 3D motion appendage. We're going to make a few modifications. The first one is that we're going to vary the area throughout the time that we're estimating these forces by using angle zeta and gamma that we get from our shrimp kinematics. So the area is going to be a function of these two angles. we're going to estimate the coefficients. Drag and inertia coefficients are extremely difficult to find for very specific cases. They're usually available for cylinders and spheres and whatnot, but very difficult for appendages that are moving about. So we're going to estimate these using the analytical solution for an oscillating plate, also called the Stokes-Wang solution. And I'll spare you all the details there unless you really want me to tell you. But our coefficient due to inertia is going to be between 2.1 and 2.2. And our coefficient due to drag is going to be between 3.5 and 6.5. We'll also get the velocity and acceleration for our equation up there from the Schirm kinematics by just taking the displacement over time. In the end, this is the equation that we get. The coefficients there are going to be different from the coefficients at the top. That's because instead of an oscillating plate for which this equation is modeled, we have a rotating plate that's also taken into consideration. And here I show you the results right away. We have our model and our experiments. And you'll see that these are extremely close. I think for the planetary people, this is within 4 orders of magnitude, no problem. So you're good to go. These are within the same order of magnitude. There's about a 30% difference in the case that is most off. But as you can see, we can really nicely estimate the forces on this appendage using this 1D equation that only has 2 force contributions to comparing to our 3D motion. We can also catch the landmarks in this force. So we're going to have 2 peaks and 2 troughs. We can estimate those using this very simple equation as well. So that is really important, not just for the engineering side, but also for the biology side. Biologists tend to not have access to particle image velocimetry or be able to do direct force measurements. And if they have kinematics of their animals while they're swimming in the ocean, they can use this very simple formula to estimate the forces and kind of understand the propulsion mechanism for the animals. I am still trying to answer some questions about this system, and those include understanding the contribution of this leading edge vortex to the total lift, which will not be 100%. So I'm still very much working on this as well. So really now we're going to end, I guess, the most fun part. The next part is also fun, but might not be as fun. But I hope that I've shown you that we can achieve both biology-inspired engineering and engineering-enabled biology through understanding the fundamental principles of shrimp locomotion. And this can both inform the creation of new underwater robots, as well as help us understand the ecology. I know I'm just going to need to drink water like halfway through this. And I know I talk about shrimp a lot, but really, what really is interesting about shrimp the most is their fluid dynamics, right? And fluid dynamics are ubiquitous; they're everywhere. And if you are as distracted as I am, you're going to find it and be able to do experiments and find things in all sorts of places. And I've really been able to do so. So we've been able to study copepods. They're small organisms that live in the ocean that perform neo-vertical migrations in the water column, and how they can reverse their near body flow to take advantage of gravity, which is really interesting. I've been able to help the swim team at Brown be more efficient by understanding their kinematics, by taking videos of them swimming. obviously more shrimp, of course. And I've also done something that would really at first sound out of left field, which is work on the bioremediation of lunar regolith simulants. And because I think that that would be of most interest to this crowd, I kind of want to go into it just a little bit. The preprint is on bioRxiv, so if I'm not giving you enough details about this work, please go check it out. But really, what we're trying to achieve with this is use lunar regolith or the moon dust to plant on the moon. So having horticultural crops in this medium. And to be able to use this medium that is toxic, we need to first bioremediate it. And we're doing so through different methods. First, we're using arbuscular mycorrhizal fungi and vermicompost. We've been able to show that by using these two methods, we can sort of bioremediate this medium, really turn it into a medium and grow chickpeas in it. And in fact, we were the first to be able to grow chickpeas to seed in lunar regolith simulant. The seed that you see right there is the first seed to come out of these plants. And from the fluid mechanics perspective here, what you need to understand is that this lunar dust is really, really fine, and there's not a lot of interstitial spacing between these particles. This makes it really hard to water the plants when you need to be watering plants in this medium. So now we're not top watering, so we're not doing as you would do with regular plants, you know, pouring water. At this moment, we're using a cotton wick, but what's really interesting is that this arbuscular mycorrhizal fungi secretes this protein that agglutinates or aggregates your lunar regolith simulant. And we show that here in this plot where you have different concentrations of this substrate that have been inoculated with this fungus and non-inoculated. And what the fungus does is aggregate these particles and in that way create better pore spacing for us to be able to water. So it's really a work in progress. I'm doing this with a team at Texas A&M. And if you want to learn more, feel free to reach out. Yeah, now I will try to speed talk my way through this. These are the three main projects that I'm trying to accomplish during my time here. I am going to focus on two of them for this talk. It's really in the beginning stages, so please be patient as I sort of work through this. But if you have any interest in working in any of these things, or if you have any questions, please reach out. I am conducting this work at this time, so I'm always happy to have someone else come into the project. The first project that I'm working with in collaboration with UT-MSI down in Port Aransas is understanding mussel and oyster reef flow. I won't talk so much about this project, but it's really interesting. It's going to take advantage of the facilities at Port Aransas and the National Reserve that they have there to conduct this work. Here, I am working on two main projects. So this reduced gravity sediment transport, and if you know Perry's work, if you heard her talk, I think 6 months ago, you'll understand why I'm doing this. Perry is doing the modeling side of understanding erosion rates in ocean worlds. And those are really based on models and empirical equations. And I want to simulate those conditions in the flume here at UT and be able to better understand what's happening flow-wise. And once again, the flume right in the building next door. If you want a tour, I have promised a tour to several people. So I'm happy to do it. And the second project that I'm working on are these geometry-dependent tracers. And I'll go into what this means, but sort of my vision for this project is what you see right there at the top, where you have different tracers, just particles that have different shapes. And due to their shape, they can capture different flow features. And these can be used in areas that are very difficult to access, such as the marine terminating glaciers during the winter. And I'm working on this with Benjamin. So, and I'll quickly go over this, but looking for at sediment transport from more of a fluid dynamics perspective, we can look at the particles and do a force balance on these particles where you have lift and drag, and you have the submerged weight. These are the equations that we're going to use. We have our little force balance there. And really just a quick summary of what we can do with this. We can use this force balance to estimate the saltation trajectories of particles as they are being transported in this medium. And this plot is going to show you the comparison between the three of them. So if you're looking at Earth, Europa, and Enceladus, they're going to have different gravity constants. And for that reason, the particles are going to have different behaviors in these three different flows. So when you look at this problem with the same initial conditions, you will see the wildly different behavior that you have. When we're talking about these particles being transported on Earth, you have lengths in the range of centimeters. But when we look about these particles in Enceladus and Europa conditions, you have saltation lengths of meters. So I'm going to be doing experiments to further understand this and also to understand what else we need to consider when we're doing this force balance to have a realistic prediction of the saltation trigger. So I'm going to do very simple, but not easy experiments where I have saltating particles in the flume, and I'm going to be tracking these particles, measuring some values of interest, and then inputting them into this model to then validate and further understand erosion rates. The second project I'm working on, which I think is quite exciting, are these geometry dependent Lagrangian tracers. And once again, I'm going back to bio-inspired. I had to find a way. And this is inspired by how phytoplankton behave in currents. So phytoplankton, you're going to see a range of different phytoplankton in this image. The scale bar is 20 microns. So these are very, very small organisms. And they have all sorts of different shapes. Most of them are not motile, so they just ride the currents, and they don't really have a choice but to do that. So what's interesting is that they can take advantage of their shapes to ride different types of currents and to sample different types of flow in the ocean. That's sort of what I'm showing here. And what has been shown is that plankton that are elongated, they have a much better chance sampling the upwelling region, whereas plankton that are spherical are much better at sampling the downwelling regions. Additionally, with spherical plankton, you have a lot of clustering of this plankton, but elongated plankton does not cluster. So really, there's nothing that differs from these two situations rather than the geometry. So can we take advantage of the geometry to design tracers that can sample different types of flow without needing to be activated? And this is really the idea and where we want to go. Where we're at right now is that we are making these tracers. I have a 3D printer in my office if you need anything 3D printed. I just wanted to put that out there. And so far, we're trying to understand how you can have these tracers at the bottom of the ocean for different amounts of time and then recover them. And this is sort of in that list, design parameter, time dissolution. Can you have a time dissolution that this particle, part of it will disintegrate and will rise up after a predetermined amount of time? This would allow us to do, for example, a chemical profile that is depth integrated. So you could drop this tracer in the ocean. After a certain amount of time, it'll rise up and it could collect samples in all sorts of ways. So the current progress on this is that, yes, you can design tracers that dissolve at different rates. And this is the video. So there's lead at the bottom. And once this part dissolves, the lead is going to drop out and you're going to be able to recover your tracer. And this happens within the span of an hour. Of course, this is just really the beginning of this project. It is supported by an innovation award here at UTIG, and there's much more to come. We're just starting with spherical particles. We're going to examine different shapes, see how they behave, and then it's really a matter of connecting the behavior of these particles to properties that we want to measure using traditional methods, and then creating a relationship between these. So to really end this, there's a lot of people that contribute to the work that I do. So many people at so many institutions, and I always want to acknowledge that I never do anything by myself. Well, I do everything myself, but you know, there's so many people around me that have brilliant ideas and are so helpful and supportive. My robot work was funded by NASA, and the work that I'm doing here on the geometry is funded by that innovation award. And I want to leave you with that little jumping shrimp. It's only 2 millimeters. And it jumps out and generates these vortices. So I'll take any questions.
Hi, that was fun. I loved hearing about the shrimp, and I'm going to ask about the shrimp. So,
Okay, sure.
I'm curious about the robot, and we kind of got a sense in that one video you showed of you like putting it in the water how big it was, but I'm curious about like, okay, so shrimp are fairly small, and you're building this robot. How big is the robot like relative to shrimp, and does making the robot bigger or smaller affect the way that like the fluid dynamics works?
That's a great question. I'll try to answer it by parts. So it really depends. It really depends on what shrimp you're comparing it to. That's always my first answer. If it's a tiger prawn, they're yay big. So they're invasive in the Gulf of Mexico. They taste delicious. So if you can catch them, they're yours. But yeah. So this robot shrimp at this moment is much larger than we would like it to be, about 10 times as big. The point of these is that we'll be able to put them into capsules and then release them into the ocean. So we'd want them to be much smaller. We don't expect that the size is going to change the fluid dynamics, because we see that shrimp from 2 millimeters to 15 centimeters swim the same way with the same resulting fluid dynamics. They do change their kinematics though. So if we want to scale this robot down or up, we're going to change the kinematics. That's actually quite simple to do. Does that answer everything?
Yes, thank you.
So I guess you just mentioned that there's a bunch of different types of shrimp, and you showed the benthic and the pelagic.
Yeah.
Do you hope to, I guess, change the locomotion of the robot if you're doing a study in the benthic region versus the pelagic region, or do you actually have to manufacture like a separate robot for that?
No, you don't have to manufacture a separate robot. That really comes down to controls. So the two videos that you saw, the comparison, they're exactly the same robot. They're just different amplitudes. And that's really all you need. We've found that even if you want to turn this robot, really all you need is to have a different amplitude on one row rather than the other, and it'll turn very easily. So that's the nice thing about shrimp, is that they always use the same propulsion mechanism. Even when they're turning, everything is happening the same. So they're a great model organism because it only comes down to control and not actually manufacture different design.
I had another quick question, I guess, just looking at shrimp.
Yeah.
And your robot, lots of moving parts.
Yes.
How how has that been challenging, I guess, for scaling things and making sure that this locomotion actually is coordinated? So I would imagine that's really challenging.
Yeah, it's really challenging. That's why it's taken, 4 years to get to have a really small robot. At first, every part was sort of thought about independently. So every part had its role. And now that we've moved on, each part has like 10 different roles. And we do so by designing it really intricately. So if you have a bendy motion, instead of having something control it, you're just going to have perhaps a plastic shim that is going to bend one way and not the other. So that's been very challenging, and making it smaller and smaller. It has been very difficult because as you reduce your tolerances, as you try to put more mechanisms into this small design, it gets very challenging. So now we're trying to move from servo actuated to solenoid actuated, which will make it much easier, and studying different ways in which we can design for function. So instead of having something that is bendy or having two parts that bend together, having one complete part that has grooves that allows it to accomplish the same thing with much smaller number of parts.
Great, thank you. I have a question regarding, so on the shrimp, you have those pleopods, the endopodites, exopodites, right?
Yes.
And other shrimp, you also have those kind of filaments attached to the pods. Do they have any function for the shrimp?
Yeah, so the filaments are sort of, in the fluid dynamics perspective, they're an extension of the membrane. So they function as a complete membrane. There's not much fluid going through it, but it allows shrimp to sort of retract their hairs during the return stroke, and create a much smaller area. So we think that's sort of the function there. I did look into it, and the difference between you having a full paddle that has no porosity and having this very ramous filamented paddle is about 90%. So 90% of the flow will still be redirected even with the filaments. So we don't think there's a ton of fluid dynamics difference, but there might be ecological and morphological differences there.
Thank you for that talk. That was so cool. I was wondering if your recent work here on like sediment transport in reduced gravity environments have informed any opinions on what you think the optimal size shrimp is for like a Europa or Enceladus environment.
Not yet, but that's definitely something I'm also looking at, because that's actually what I proposed here for the fellowship at UTIG, was to understand the scaling of robots, because you do see a large range of really small robots, really large robots, and can they perform the same functions in different environments? So that's definitely coming, maybe a little bit further down the line, but definitely coming. So thank you for the question.
Thanks so much for the talk. Although it's way different from what I do. So I have one question. Because we know the shrimp are very small.
Yeah.
And from what they eat, and how they swim, what's the energy ratio between that? Like, I know it's elegant way of swimming style, like providing the thrust. Is it like a very efficient way of doing that? And in terms of design, like a robot, like mimicking their swimming style. I guess it's one thing to consider in design. You want to be efficient, right? You want this to last longer, considering a limited battery capacity.
Yeah.
That's
So shrimp themselves are not the most efficient swimmers. They are extremely maneuverable. If you want the most efficient swimmer, it's actually jellyfish. They just contract their bell. That takes no energy or very little energy. Shrimp have the advantage of being extremely maneuverable. So that's why we're looking at them. They're not inefficient in the way that they swim, but they're not the most efficient if you look at all the animals. But they really are one of the most maneuverable. They can always also swim about 10 miles a day horizontally, which is a long distance for their small size of 5 centimeters. So we think that even though they might not be the most efficient, there definitely are some advantages to having shrimp as your model organism.
Thank you for the talk. That was
Yeah.
really fun. So most of your results were normalized by the period or the frequency of the oscillation. But I wonder if these things are frequency dependent, like say the lift or the thrust. Have you thought about it?
Yes, I personally have not looked into it, but there have been teams that have looked into the frequency. And if you sweep through frequencies, or if you sort of sweep through this phase lag, because the frequency will sort of be also dependent on this phase lag. Shrimp have the most efficient phase lag when it comes to transporting flow from the beginning of their appendages to the very last of their appendages. So that's sort of where we left it, but I haven't really looked into it that much.
All right, thanks. We wrap up the questions, but note that Sarah is local, so you're very welcome to email her to chat more. And there's also a schedule that you could sign up if you want to meet today. There's still a couple of openings in the afternoon. So thank you again, Sarah,
Yeah,
for a
thank
great
you.
talk.